I hope the slides are visible to you. If you cannot see, please let me know in the chat box. I am going to start today's session then. So in today's class, we will mainly discuss about the questions related to carbohydrates. You have already submitted the assignments regarding that, so I can discuss those questions as well. So if you have any doubt during any question, you are free to ask during the, or you can ask your question in the chat box. Okay, so let us begin. So this is the first question of today's session. So question number 1 is stating that the main component of the plant cell wall is what? So this is your question. There are 4 options. A is cellulose, B is glycogen, C is peptidoglycan, and D is lignin. So these are your 4 options. You have to select what is the main component for plant cell. I think all of you already know this answer. If any of you can, please write your answer in the chat box and then I will show you the correct answer for this one. Okay, fine. So I don't see any answer yet. So the correct answer for the question should be option number A, cellulose. So cellulose is, one second, let me just write it down. Cellulose is a beta 1,4 linked glucose and it has an unbranched structure. During the classification of different carbohydrates, according to their roles, we get to know that there are mainly 2 types. There are actually many types, but depending on their role, some can actually act as storage or energy storage, or some have some structural roles. Among them, cellulose does have a role for structure formation. So cellulose, because it is having a role for structure formation, it is an unbranched structure, and in case of cellulose, the polymers or the polysaccharides are actually linked by beta 1,4 linkages. And also, so you can think it like these are parallelly branched structures and they are highly interlinked by hydrogen bondings. This gives a very firm structure to this. That's why it is used for the plant cell walls. Building the plant cell wall, that is why it is used to give it a firm structure. So the correct answer for question number 1 should have been cellulose. Okay. Other options you already know. Glycogen is something that is found inside animals, and peptidoglycans are the things that is made the bacterial cell walls. Moving to the next question, it states the main functional group present in carbohydrates are. So there are 4 options again. So you have to identify what are the main functional groups present in case of carbohydrates. Please do try to write your answers in the chat box. Meanwhile, I am explaining. So we are dealing with carbohydrates. What are these? These are oxidized polyhydroxy alcohols. And what do they contain? They often contain, yes, I can see one answer. Thank you very much. Yes. And what do they contain? They often contain either aldehyde group or ketone group. Where is this coming from? Because if you remember, the parent compound for carbohydrate was glycerol. This was the structure for glycerol. Now, this can lead to the formation of 2 main parent compounds. These are, one of them is conversion of this CH2H to CHO, leading to the formation of glyceraldehyde. Sorry, let me just write it down here. This is your glyceraldehyde. And another one is CH2OH, CH2OH. This is your dihydroxyacetone. In both the cases, what are commons? You can see these are the parent compounds that are, or you can say the parent building blocks for carbohydrates. So for all of them, you can see the main things that are common are the hydroxyl group, as well as you can see there is a CHO group, that is aldehyde, that means you have a carbonyl group here as well, and a ketone, that means here a carbonyl group as well. So the correct answer should have been hydroxyl groups and carbonyl groups. The most of you have answered aldehydes and ketones. Yeah, yes, exactly, aldehyde and ketones are a major part of carbohydrate, obviously, but hydroxyl groups are the main part for carbohydrates as well. That thing is not included for option number D, because if you only say aldehyde and ketone, I can make aldehyde and ketone out of anything. Like, see, if I have aldehyde here, but let's just continue the chain like this. This is also an aldehyde. Again, we can have our ketones like this. This is also a ketone, simply like acetone. But is it a carbohydrate? Is it just checks all the criteria for having the carbohydrate? No. Why no? Because the hydroxyl groups are an important part. They are required for further cyclization of these carbohydrates. Whenever you are going to the pentose sugar and aldose and ketose sugars, in all those cases you need the cyclization or the hemiacetal formation. For those, you both require a carbonyl group where the attack will happen, and OH group or hydroxyl group that will do the attack. Because in all the cases, whether you are forming a furanose or a pyranose, whatever you are forming, in all the cases one hydroxyl group attacks the carbonyl center. This can either be an aldehyde or this can be a ketone as well. So the correct option for question number 2 should have been option number A, hydroxyl groups and carbonyl groups. I hope this clears your doubt, because most of you answered D. Okay. So let us move to the next one. This is your question number 3. It is stating which among them which of the following is not a hexose sugar. Hexose means, obviously you know, hex means you have 6 carbon atoms. These are the options: glucose, fructose, ribose and galactose. You have to identify which among them is not a hexose sugar. So for this question, okay, I already have an answer. Exactly, you are absolutely correct. The answer that you gave, yes, ribose is not a hexose sugar. So in case of the options, what is a glucose? Our glucose is a aldohexose. Sorry for my bad handwriting again. Fructose is a ketohexose. Ribose among them is a aldopentose. And galactose is also a aldohexose. So first of all, what does this mean? Just as I said, hexose means you have 6 carbon atoms. Pentose means you have 5 carbon atoms in this moieties, in this sugar moieties. And aldo and keto means whether you have an aldehyde group at the top or a ketone group at the top. That is what it means. So only among them ribose is something which has 5 carbon atom. So our correct option should have been option number C, ribose, which is a aldopentose. Okay. Sorry for that. Yeah. We just discussed this question. Which among them is a ketohexose? Just in the previous question, I have already written down the answer that fructose is a ketohexose. So obviously this answer I am not going to ask, because obviously I just showed in the last question. So the question was, which among the following is a ketohexose? The option was fructose. So again, as I was in the last question I was explaining that what is the difference. With this picture it will be more clear to all of you. So based on the number of carbon atom, we either write it as hexose or pentose. So how many carbon atoms are here? This is the structure for D-fructose. So how many carbon atoms are there? 1, 2, 3, 4, 5 and 6. So there are 6 carbon atoms. So it will be a hexose. And here you can see, instead of a CHOH group, we have a COCH2H. What is this? This is a ketone, this part. Instead of that, you can have CHO. Whenever you will find this functional group at the top or a aldehyde group at the top, it will be called as aldo. It can either be hexose or pentose. But in this case, this is a keto group, so it is called as a ketohexose. So depending on that, we are classifying them. And the next step that is shown here, it is the cyclization process. Just at the beginning of the first or second question, just I showed that you need a carbonyl center and a hydroxyl group for further cyclization. Whether you are forming a furanose or a pyranose, whatever you are forming, you need a carbonyl center and a hydroxyl group for the attack to occur. So in case of D-fructose, how the cyclization occurs, that is shown here, with either this 5-OH group can attack and lead to the formation of this fructofuranose, or this 6th position OH group can attack here and form this D-fructopyranose. Any of them, either any of these two can form. Okay, let us move to the next question. It is saying the reference compound for naming D and L isomers of sugar is what? So we can classify sugars, or we can based on their stereochemistry, we can classify them whether it is a D isomer or a L isomer. So you have to identify what is the reference compound based on which this nomenclature has been derived. So option A is glyceraldehyde, B is ribose, C is glucose and D is alanine. So you have to select the correct option. You can write your answer in the chat box if you know it. So based on what the initial classification of D or L was done? That is your question. Okay. Yes, absolutely correct. The correct option for this is glyceraldehyde. So what was our thing? You have a CHO group, you have a HOH and a CH2OH. Again, I can write this structure as CHO, HOH, CH2OH. So these are the two isomers that I can draw for glyceraldehyde. So what was the idea behind D and L nomenclature? That whenever you can see for glyceraldehyde, this was the chiral carbon atom. This is your chiral center. This one is your chiral center. So what does this chiral center means? That in case of carbon, you can see there are 4 different substituents or 4 different functional groups that are attached to it. Only in that case we call it a chiral carbon or any chiral atom. But in this case, because of these 4 different functionalities, H, CHO, OH and CH2OH, we will call it a chiral carbon. So in that case, we have found our chiral carbon, and if the hydroxyl group is on the left hand side, then we will call it L, and if the hydroxyl group is on the right hand side, we will call it a D isomer. That is was the basic nomenclature. Obviously you have to first put either the aldehyde group or the keto group, whether it is a aldose, whether it is a ketose, you have to put the aldehyde or the ketone group at the top, then do the Fischer projection, and after that you have to find out the chiral carbon. In this case only one chiral carbon is there for glyceraldehyde, which was the reference one, but we already know for glucose, fructose, everything we have more number of carbon atoms. In that case what we do, we put either ketone or our aldehyde group of the top. We have our carbon chain going. I am not writing it fully. The farthest chiral carbon that you find, you identify that. Suppose this, I don't know how many carbon atoms there, it can be 4, 5, 6 in between, but you have to find out the farthest one from this aldehyde or ketone group, then see whether the OH group is on the right hand side or left hand side, and depending on that you can mark it as D or L. So okay, all of you those who answered option number A was correct. Let us move to the next question. It states, to form furanose, the hydroxyl group from which position of an aldohexose attack the carbonyl carbon. So here we are talking about cyclization and we are forming a furanose. So what is your furanose? Furanose means a structure that resembles furan. That means formation of a 5-membered cyclic ring. This is your furan 5-membered cyclic ring. So you are forming furanose in case of aldohexose. So from which position the hydroxyl group will attack the carbonyl carbon? This is your question. Please do write it in the chat box, and then obviously I will discuss the question in details. Okay, we have one answer. One answer, I guess. Yes. So let us see. First let us draw the structure for that. So after drawing the structure, let's mark these carbon atoms. We know these CH2OH groups always comes at 6th position. This is 6, this one is 5, this is 4, this is 3, this is 2, this is 1. Now the attack will happen to this carbonyl center, and what you have to form? A furanose. That means a 5-membered ring needs to be formed. So let us see where should the attack go. This is your 1 carbon, this is your 1, 2, 3, 4, 5. Now this has to be cyclized. So this is your first bond, 1. This one is your second one, 2. This is your third one, 3. This is fourth one. Now if the attack happens from here, let me just show it. 5, 4 prime OH group. If the attack happens from here, one second, my pen is not working. Okay. So now if the attacks happens from here, we will end up with a furanose ring. How it will look like? So now the structure will look something like this. Just vaguely I am writing. So you had one oxygen atom attached here. You have another oxygen which has attacked to this carbonyl center. Now you have your OH group, hydrogen. This already had an hydrogen atom like this. and this is your CH2H group. Okay, so fine, I should have been the structure. Let me see whether I have it here or not. Then it could have been, I don't think I have the structures. Then it would have been much more prominent. Anyway, if you can see the structure, you should already understand that the attack should happen from this particular oxygen atom, which is situated at C4 or the fourth carbon atom, and after the cyclization we will end up with this one is your first bond, this one is second, this one is third, this oxygen one is your fourth bond, and after this attack it will form this fifth bond, forming a 5-membered ring here, which is your furanose. So that will end up to your formation of furanose. So yes, the answer that Shrishti gave was absolutely correct. Okay, so let us move to the next question. It states, among the given option, which is a multi-branched polysaccharide of glucose that served as a form of energy storage in animals? So I have discussed this question already. So we have basically two storage proteins, one in case of plants and another in case of animals. So in case of animals, we have this glycogen, which is a multi-branched polysaccharide that serves as our energy storage. Now in case of any kind of storage carbohydrates, you know the structures are generally branched because you need to store much more information or much more energy inside that. That's why you have a branch structure. Now in case of, as we already know, just let me write it down here, glycoproteins. So we have our storage proteins here, storage carbohydrates. They can be classified for plants or for animals. For plants we have starch, and for animals we have glycogen. So both of them are branch structure, and their structure looks something like this. Let me just draw it here. I am not drawing the exact structure but just a representation. So these are your branching on the side. Now in case of both starch and glycogen, you will have this branching. The only difference is that the main change that you are seeing here, the linkages between the monosaccharides is alpha 1,4 for both the cases, and for the branching units the linkage is alpha 1,6 linkages between the monosaccharides. The only difference is that how many number of branchings will be there and how many glucose units will be there in the branching. That will define whether it is the structure of a starch or it is a structure for a glycogen. Otherwise, the basic structure for both plant storage carbohydrate and animal storage carbohydrates is same. The only difference is that how many number of glucose units are there in the main chain and how many branch chains are there and what are the glucose units number in the branch chains. So for question number 7, the question was simple. I just discussed for the other things for your understanding. So the question was very simple, that among all these options, which acts as a energy storage for animals, I have already written down the answer, that is glycogen. So the correct option for question number 7 should have been option number B. Okay. Moving to next one. Okay, we just discussed this. Starch and glycogen are both polymers of what? So we just discussed it, that they are storage protein. They are polymers of glucose, and what is their form? This is alpha D glucose. In both the cases, they are alpha D glucose. If you remember your lecture material, it was already said that in case of nature, between the two isomers, D isomer and L isomer, in nature, you will always find the D isomer. In all the cases, you will see the carbohydrates that are present in nature are D carbohydrates. And we just discussed in the last slide that in all the cases the linkages are alpha. See, in the main chain the linkage was alpha 1,4 and in the side chain was alpha 1,6, both in case of starch and glycogen. So that means in all the cases for storage carbohydrates the linkage is alpha. So for this question, that starch and glycogen are both polymers of what? They are both polymers of alpha D glucose. So the correct option for this should have been option number C. Let us move to that. So it is saying which of the following is an anomeric pair. So this is already written here. I think you all know the answer. Please do write it in the chat box and then I will explain it. Which of the following is an anomeric pair? I have two answers. Yes, absolutely. The correct option is option number C. So what are anomers? So first of all, the option number 1. Okay. Option number 1. So who has a question? Someone raised hand. You can write your question in the chat box. I am constantly reading it. So if I know it, I will be able to answer it. Okay. So coming back to the question. What were anomers? So anomer means you deal with a single sugar or carbohydrate. Now in the first case, anomers are the isomers of a single sugar or single carbohydrate. They are isomers. That means we are dealing with the same sugar. But in case of the option A, we have glucose and mannose. They are completely two different sugar moieties. So obviously this option is incorrect. In the second option, we are dealing with alpha D glucose and beta L glucose. We are still considering it. The answer I have already showed, but for the sake of explaining, we are still considering it because still we are looking for at the same sugar, that is glucose. Third option was again about glucose and the fourth option was again about glucose. So among these three we have to choose our correct option. So what is the difference between anomers? See, if we write down the structure, let me just write it here. So we are talking about glucose. This is not going good. Just let me write it down here. Another one is, I am not writing the whole structure, just the bit of it which I will require to explain. CH2OH. There are other functional groups as well. I am just writing it. This is CH2OH. You have your CH2OH of this here. So we can write down actually glucose in two different forms. One is alpha form and another is beta form. Now in case of these two structure, this one is your alpha form, this one is your beta form. So when do you call something an alpha form and when do you call something an beta form? Whenever this one is your 6th carbon atom, this is 5, 4, 3, okay, this is pyranose 2, I have a missed one, yes, I think I mixed here, 4, 3, 2, just let me write it down, 2, 1. One second, let me change the color, it will be easier for you to see. Okay, so this was our 6th position, this was our 5th position, this was our 4th position, this was our 3rd, am I missing one, 3rd position, this was our 2nd, and this was our 1st position. Just note down the green ones, do not look at the red ones. I missed one during the marking, just let me cut them down. Just follow the green markings. So this one is your 6th carbon atom, this one is your 5th carbon atom, this is 4, 3, 2, 1. We have our glucose sugar. I am not writing all other functional groups that are present, just writing the CH2OH group and the OH group at the 1 position. So whenever you see the CH2OH and the OH group at the 1 position are at the same site. Now in this structure, both of them are axial position. That means both CH2OH and OH groups are pointing towards the same direction, or we can also call it as a cis, because they are both pointing towards the same direction. In that case, we call that as a beta structure. And in case of this one, you can see this is your 6th one, this is 5, 4, 3, 2 and 1. So this 1 position's hydroxyl group and the CH2 group are not in the same direction. This CH2OH is in the axial position and the OH group here is in the equatorial position. So that means they are not in the same direction, they are in the opposite direction. That means they are known as alpha. So whenever you see the CH2OH group and the OH group at the first position are in the same direction or in the cis direction, we call them as beta, and whenever they are in the opposite direction or in the anti direction, we call them as alpha. This structural difference between glucose or any kind of sugar is known as the anomeric structural, or they are both together known as anomeric pairs, where you can draw them either as cis or as a trans. D and L is a different isomer than alpha and beta. You have to be very careful about that, because in case of D and L, it is originating just as we have discussed, that you put the aldehyde or the keto group at the top and you find out the furthermost chiral carbon atom, and then find out whether the OH group is on the left hand side or on the right hand side, and depending on that you mark them as D or L. But in case of alpha and beta, you have to look at the CH2OH group and the OH group and where they are pointing. If they are pointing at the same direction or in the cis positions direction, they will be beta, and if they are anti, they will be alpha. So that was the thing. So let us see. I think someone answered it and that was correct. Okay, let us move to the next one. This question is stating the bacterial cell wall is made up of what? I have already said it, I think, in some questions, so I think you all know. So what is the bacterial cell wall is made up of? These are your 4 options. Please write your answer in the chat box and then I will show you the structure in details. Exactly the correct option that you have said. The correct option is peptidoglycans. We have already gone through this when we were understanding the action of lysozyme, that in the bacterial cell wall you have alternating NAM-NAT units. What is NAM and NAT? You have your N-acetyl glucosamine and N-acetyl glutamic acid, they are alternated. So here, one second, here if you see this picture, you can see the alternating NAM-NAG units are shown here. Another important thing, this is not the question that is asked here, still I would like to say that these are actually unbranched structure. In this polysaccharides, you do not see any kind of branching that is coming from other sugar moieties. This is a simple chain. See, here is another chain, here is another chain. This is unbranched structure, but you can see there are some bondings between them. In all those places you can see some bondings. Let me just show it like that. So this is your alternating NAM-NAG units. This is one strand, this is another strand, this is the third strand, and this how it will continue. And you have some other units in between. So what are these things? These are cross-linking. This is, you should not confuse it with branching. Branching is different. If this have been a branching unit, then these all the round things that you are seeing, they will also look like hexagons, because we are talking about the hexose sugars here. So if the branching was also made of sugars, then the branching, the spherical units also would have been hexagons. But these are different. These are other cross-linking. These are peptide cross-linking. You have to be very careful about remembering this structure. This is very important because bacterial cell walls are actually very heavily cross-linked by peptide bonds. This here you can see many names are here written as. These are peptide cross-linking that you can see, and that gives the bacterial cell wall their strength. So again, see, in case of structural matters, we said previously as well, whenever carbohydrates have a structural role, they do not go for branching. They only go for branching when you need to store something, store information, store energy. Only in those cases they show branching structure, like what we have seen in case of starch and in case of glycogen. But whenever they have a structural role, like in the plant, in case of plants, in case of cellulose, in case of plant membranes, you have this cellulose cell walls. So in that case also you had those unbranched polysaccharide units. Here also in case of bacterial cellular units, you have this unbranched peptidoglycan units, which are heavily cross-linked through peptide bonds, and that is why the bacterial cell wall is very difficult to go through, because obviously the bacteria will try to survive. They will try to survive inside different organisms. So obviously their cell wall has to be very strong. They do not want anything from outside to get through them and kill them, obviously. This is why the structure for bacterial cell wall is very strong and it is very difficult to break it. Okay. So yes, the option was C. Let us move to the next question. Oh, a similar question we have already discussed. The hydroxyl group from what position of an aldohexose attacks the carbonyl carbon to form a pyranose? So in the previous question we were asked about furanose. Now in this question they are asking about, one second, yes, they are asking about pyranose. Pyranose means you have a 6-membered cyclic ring containing a oxygen atom. That's why they are called pyranose. So here the same question again. From which carbon atom the hydroxyl group will attack to form a pyranose? Please write your option and then I am showing you. So let me just draw it here. Yes. So we had a structure like this, which we have H, which here we have our AHOH, and this is like CH2OH hydrogen. So from which position the attack is occurring, let us see. I already have one answer, still let us see. First let us do the markings. So this is our one carbon, this is our second carbon, this is our third carbon, this is our fourth one, this is fifth one, and this is our sixth one. So for formation of a 6-membered ring, you can already see this is our first bond, this one is our second bond, this one is third, fourth and fifth. We already have our 5 bonds. Just an additional bond formation will result in a pyranose formation. So now if the hydroxyl group from this position attacks here, you will end up with a bond generating here, and that will lead to the formation of pyranose. So from what position the hydroxyl group is attacking? From C5. This is your fifth carbon. The OH group is attached to the C5, the fifth carbon. So from that position the attack was happening. Yes, the option that you chose is absolutely correct. Is option number C. From C5 position the attack will happen. Okay. Coming to the next question, it states carbohydrates. There are 4 options. You have to select which of them are true. They are first option is they serve as a structural element of cell walls for bacteria and plants. Second one is play key roles in cell recognition. Third is act as energy storage, metabolic intermediates, and the fourth one is form part of structural framework of RNA and DNA. Out of these 4 roles, which are true for carbohydrates? You have to find out the true options out of these 4 statements. Read them very carefully and select your option accordingly. And then I will discuss the statements one by one. Just go through them and then select your option one by one. Which of these statement 1, 2, 3 and 4 are correct, and accordingly select your option from below. Okay, so let us look at the first statement. It is saying carbohydrates serve as structural elements of the cell wall of bacteria and plants. So just a few question backs we already know. Yes, I already have an answer. Thank you very much. Okay, so we already know they do act as structural elements for bacteria and plants. In case of plants, we know that about cellulose, and bacteria, it is peptidoglycans, they form the cellular wall. So yes, the first statement about carbohydrate is a true statement. Coming to the second one, play key roles in cell recognition. So cell recognition, you can think it something like that, if suppose this is one of your cell and it is trying to configure its neighboring cell, whether it is a same type of cell or not. So sometime what can happen, say in your suppose in the neighboring site you have a disease cell or some kind of disconfiguration, something happened in that cell, so that is not a properly normal cell. So you need to identify that, the cells need to identify, because otherwise if the cell have a defect and they do not identify that, then that defect will continue. If the cell start progressing and cells start dividing, then that defect will continue in your body. So the cells, it is very important for the cells to actually identify the neighboring cells around it, and sometimes also they transfer information from one cell to the another. So this is something like a cell cell interaction. In very vague way I am just saying. So for this cell cell interaction, you can already imagine if two cells need to interact, first thing that will interact is their cell walls. So what do you have in the cell walls in case of cells is you have the cellular membranes. If this is your cell, on the outside what do you have? You have your cellular membranes, and what the cellular membranes made of? You have already known it is proteins and lipids that does form the cellular membranes. We have already discussed this many times. This bilayer type of structure, we have discussed it multiple times, that the cellular membranes look something like this. So these are your proteins and lipids, and if they need to interact, these membranes have to come to a closed space. Only then they can transfer information from one place to another cell, right? So most of the times the cell recognition, the carbohydrates that are involved in that are membrane carbohydrates. So if a carbohydrate is involved, membrane carbohydrates. So if a carbohydrate is involved in transferring some information, cellular information, they have to be present in the cellular membrane. You know, many kind of sugar moieties and lipids and fats, many things remains in these cellular membranes in the interfacial positions, and these things actually transfer the information from one cell to other. So many times this happens that membrane carbohydrates which remains in the cellular membranes does play a role in case of cell recognition. So although this was not very much discussed in the lecture material, but just for your information, that there are some carbohydrates which are known as membrane carbohydrates, and they can play a role in cell recognitions as well. So this statement, the second statement for carbohydrate, is also a true statement. Third one is energy storage. We already know that they can act as energy storage, that for plant that is starch, and for animals also you have your glycogens. So starch and glycogens, those acts as storage carbohydrates. So the third option is true as well, and the fourth option is form part of structural framework in RNA and DNA. Again, for RNA and DNA, the main thing is the ribose sugar for RNA or deoxyribose sugar for DNA. So what are they? This is your deoxyribose for DNA. Ribose and deoxyribose, again they are nothing but your carbohydrates. That is what you are dealing with when you are understanding the structure for DNA and RNAs. In one case we had a hydroxyl group at 2' position. In another case we did not have any hydroxyl group for DNA in the 2' position. But all of them were nothing but a ribose sugars. So that means they do also play an important part in forming structural frameworks for RNA and DNA. So that means all the four statements that has been given here are true. So the correct option that she wrote is absolutely correct. The correct option should have been option number B. All the four statements for carbohydrate is a true statement. Moving to the next one. Furanose sugars are comprised of what? So we just discussed it. I will not spend much time in it. So you all know we have two different type of sugars. One is named after furan and another is named after pyran. So furan is a 5-membered cyclic ring that composed and containing oxygen atom, and sugar moieties looking like this are known as furanose, and looking like this are known as pyranose. So how many carbon atoms are there? 1, 2, 3, 4, and it form a 5-membered ring in case of furanose. So the correct option for question number 13 should be furanose sugars are comprised of 5-membered rings. You all know that. So let us move to the next one. Yes. Please answer this question. This is not difficult, you already know, but carefully select the answer. I have a reason for saying that. This question you already know, I can tell you. So there are 2 blanks. This is your first blank, this is your second blank. You have to put the correct options in A, B and C. So these and these form of glucose found in again a blank. So you have to fill these things. There are 4 statements that are given, 1, 2, 3, 4. Among them, which correctly fits this statement, you have to find it out. So you already know, all of you already know the answer. So please select it carefully. There can be a mistake while selecting the answers. Otherwise, I know you all know the correct answer, but as there are 4 statements, you can get a bit confused. So please select it carefully. Fill the blanks for A, B and C, and then I will discuss the question in details. Okay, so I have one answer. So let us see. So she is saying option number D. Yes, you are absolutely correct. So first let us see statement number 1, whether it fits this sentence or not. So first statement is, in place of A we have to put starch. So starch is a polymeric form of glucose. Absolutely, we just learned the starch is a polymeric form of glucose, where we can see different branching. In the main chain we have this alpha 1,4 linkages, and in the branching we have this alpha 1,6 linkages. So obviously starch is a polymeric form of glucose found in what? Found in case of plants. It is a storage carbohydrate for plants. So yes, statement 1 fits correctly. Let us look at the second statement. It is saying starch again. Starch is a monomeric form of glucose. No, we already know it is not a monomeric form. It is a polysaccharide. It is a, first of all, you have a full network of these glucose molecules which are linked to each other via alpha 1,4 linkages, and also you have branching. So this is not a monomeric structure at all. And again, for C it is written it is found in animals. No, starch is not found in animals. It is found in case of plants only. So yes, this second statement was fully a wrong statement. Coming to the third one, it is saying glycogen. So in place of A we have to put glycogen is a polymeric form of glucose. We all know again glycogen is a polymeric form of glucose that is found in case of animals. Yes, so this is a correct statement. Glycogen is a polymeric glucose which is found in animals. Let us look at the fourth option. It says glycogen is a monomeric one. This one is obviously incorrect. It is not monomeric form of glucose and it is not found in plants. So fourth option was incorrect as well. So the only correct statement were 1 and 3, and accordingly the correct option for question number 4 should be option number D, 1 and 3. Okay. Moving to the next one, it states carbohydrates that differs by their stereochemistry only at one carbon atom in their non-cyclic form are called what? Only if you change one stereo center. Change the stereochemistry of only one center. Then what do you call them in their non-cyclic form? So as an example, I am just showing you. Suppose this is our structure. AHOH, AHOH. Now you are changing the stereochemistry of only one carbon center. Suppose I am changing it to OHH. Other things are remaining same. So what do you call them? What is this type of structures known as? This pair of structures are known as what in case of carbohydrates? Changing the carbon atom stereochemistry at only one center. Yes, absolutely, the correct option is epimers. They are known as epimers. Anytime you only change one carbon atom stereochemistry, you call them epimer. Just like in this example, this was the marking 1, 2, 3, 4. Here also if we mark 1, 2, 3, 4. So this second structure, if we name them A and B, so B is a in what position we did the change? At C3 we did that change at the third carbon atom. So this is a C3 epimer of A. Okay, I think it is clear. Let us move to the next one. This question states the different stereoisomers formed due to cyclization at the aldehyde or ketone carbon are called what? The different stereoisomer formed due to cyclization at the aldehyde or ketone carbon. Select your answers and then I will say. After cyclization, what type of stereoisomers did you get? In the last case, in the non-cyclization part, non-cyclized structure, you got your epimers as the stereoisomers. Now after cyclization, what kind of isomerization do you get? Yes, we have already discussed it. They are known as anomers, because we all know after this hydroxyl group from any of the position for the ring formation, either forming it a 5-membered or a 6-membered ring, after it attacks the aldehyde or suppose a carbonyl group, anything. Here you have the formation of a OH group. Suppose for aldehyde you have your OH group and hydrogen, and after this attack, this 1' position OH group can either stay at a cis position with CH2H or a trans position with CH2H, depending on we classify them as alpha or beta, and they are known as anomers. So correct option for question number 16 should have been option A. Moving to the next one. Which of the following statements is true for the cyclic structure of monosaccharides? So you have to identify among the which statement is true for cyclic structure of monosaccharide. So you have your cyclization. So what do they form after cyclization? Whether they form acetals or they form hemiacetals, whether they form ethers or they form esters. After cyclization, what kind of structure do you get? Select your option and then I will tell. cyclic structure of monosaccharide. That means what do you have? You had your monosaccharide. One second. You have your monosaccharide. So what do you have here? An aldehyde group or a ketone group in case of your monosaccharide. And also you have a hydroxyl group that is also a part of the structures. I am not drawing the entire structure, but both this aldehyde and ketone group and this hydroxyl group are part of this monosaccharide structure. After the attack, what do you get? After this first attack, you get, if this is, let me just write as OR1, so you will get as OR1, and this is your, so this is hemiacetal, only single attack by a OH or a OR group resulting in a hemiacetal formation. And after the second attack, if another OR2 attacks here and you have your OH group removed, resulting in the formation of OR2, OR1, then this is your acetal. Acetal linkages happens in case of formation of polysaccharides. When the second attacks happens and you have the formation of the O-glycosidic bonds, that is the for the polysaccharides. During polysaccharide formation you have the acetal formation, but during the normal cyclization you have your hemiacetal formation, where you have your pyranose or furanose ring. During that you have your hemiacetal formation. So the correct option for question number 17 should have been option number B. Okay. Moving to the next one. It states carbohydrates that are optically active have the ability to rotate the plane of polarized light. Light rotated in the clockwise direction is generally represented by the symbol what? So there, read the question carefully. There are 4 options, L, R, plus and alpha. Select the option carefully. So you have the rotation of your polarized light, and if the light is rotated along the clockwise direction, how it is represented by the symbols? That is your question. Select your option and then I will discuss it. Okay, fine. So first of all, to answer this question, you need to understand what do you mean by plane polarized light. Now look at this picture here. Just let me on this leisure option. Just look at this light source. Originally, if you use any normal light source, the waves are in different directions. Obviously, it will happen. If this is your light source out of which the waves are coming, then they are vibrating in different directions. They are not all in the same plane. They are vibrating in different planes. So that is not a plane polarized light that you are seeing here. You can see from the arrows that it is in all the directions. You do not have a control here. But for the formation of plane polarized light, you have to put a polarizer here. So what does it do? Just look at this picture and you will be able to understand. that now the light is no longer scattered, or no longer the waves are vibrating in all direction, but there is a specific direction along which the waves are now can vibrate. This is the only direction that the polarizer is allowing. All other directions light are cut off by this polarizer, and you are only getting the waves that are vibrating along this direction. So now you have a fixed direction. This direction is now fixed after putting this polarizer here. Now you are putting your chiral solution in this tube. This is actually the setup for a polarimeter, through which the different angles, or how much angles they are able to rotate, or the muter angles for muter rotations actually determined. So this is actually the setup for muter rotation. So this was your light source, this is polarizer. Now the light is plane polarized light, only vibration is allowed along this plane. Now this is your solution. Here you put your chiral solution, and after this light passes through this solution, because it has some chirality, the molecules that are here in the solution, because they do have some chirality, they will rotate the plane of light again. So initially you have cut off all other planes, but due to the rotation of the molecules that are present here, this plane of light will again move to some degrees depending on the structure, or the whatever solution that is you are using, depending on that the rotation of the plane polarized light will occur. I will show it in a minute. So suppose now the deviation has happened. So let me just draw it here. It will be easier to understand. Sorry. Okay. Yes. So you had your light here. Now after it passes through the sample, say initially the direction was like that. Now this direction can either rotate like that, or rotate alongside that as well. This is your clockwise rotation, and this is your anti-clockwise rotation. This is happening due to the muter rotation that exists in the sample that you are using. So if the deviation happens along the clockwise direction, we denote it as a plus sign. And if the deviation of the plane polarized light happens in the anti-clockwise direction, we symbolize it as a minus sign. So you are observing here, you are observing in the polarimeter here through the analyzer. Okay, so now if you see, so in the polarimeter originally, every time you can see there is a scaling, like see this is your plane polarized light, this is marked as 0. Now here from the in front, if you are seeing, if the deviation happens after passing alongside this, then we call it plus clockwise direction, and if in anti-clockwise direction, we call it the minus. That is how we always see it. Okay, so fine, I have to, I think, 1 second. Just let me show it again, because I need the structure of glucose to show. So we discussed about polarimeter already. So how does the rotation of this plane polarized, or why does the rotation of this plane polarized light occurs? Because just if you remember, so this is the structure of a open chain glucose formation, but after cyclization, just as we discussed, they form anomers. One is alpha and another is beta. So these are the structure for alpha D glucose and beta D glucose. Now among these two structure, what is this pointer? 1 second. among these two structure, this is one is your beta and this one is your alpha. You can already see the alpha glucose is present at 36% and the beta form is present at 64%. Why so? To understand that, you need to go back to the chair form of the glucose. Let me just draw it again. Just the functional groups I will draw. So in case of alpha 1, we have your CH2OH. We have our OH groups, and in this group we have our CH2OH O, and here we have our OH group. Okay, so in case of chair form, we have two anomers, one is alpha and another one is beta, depending on what, whether the CH2OH and OH groups are on the cis side or on the same side or on the anti side. Now this happens that alpha forms remains as 36% in the solution, whereas the beta forms remains as 64% in the solution. Now whenever you put this glucose solution in the tube, due to this beta form, you have a rotation of the plane polarized light to a degree of plus 18.7. So if this was coming as a straight direction initially, after it passes through the glucose solution, you have a clockwise rotation, because you can see a plus sign in front of that. So if you can see a positive deviation of plus 18.7 due to this beta form of glucose. Now again, there are alpha forms present as well. They have a rotation of plane polarized light of plus 112. So the rotation is much more, something like this angle. So for alpha form, we have a plus 112 rotation. Now in the solution, we have both alpha and beta form. So ultimately, what will be the final turning of this light? How much will the light be deviated from its direction? So we had our 64% beta form, which deviates the light at an angle of 18.7. Also, we have 36% of our alpha form, which deviates it to a angle of 112. Now averaging it out will give us a value of 52.7. This is the final value that you will get ultimately, which tells us that both the alpha and beta form exist in case of D-glucose. Because if only a single form would have been existed, then either you would have get a deviation of plus 18.7 or plus 112. But you are not getting either of them. If you are able to get that in their pure alpha form and beta form, then this should have been the rotation values. But we don't get either of them, which again suggests that both alpha and beta form exist in equilibrium, and this equilibrium happens through this open chain form of D-glucose, and due to their presence, ultimately it results in a final rotation of this plane polarized light to an angle of 52.7. So just let me write it here. So neither 18.7 nor 112, we do not get any of them. Due to presence of them, suppose we get the rotation, if this is 60, then 30, 45, so something like this. If this was our plane, so we get a rotation like 52.7, like this. Okay, so I discussed all of this for your understanding, although these things are not included in the question, but it can come in case of exams or something, so that's why I discussed this in details. Okay, let us move to the next question. It says, Which among the following statement is or are false about beta rotation? So we just discussed about beta rotation. So you have to identify the false statements regarding beta rotation. So let us see what are the statements. First one is beta rotation is the interconversion of different monomers. Yes, beta rotation is interconversion of different anomers. You have your alpha and beta form. These are the anomers, and due to their interconversion, this is what we call beta rotation, going from alpha to beta through a open chain conformation. So our first statement was a true statement. Fine. Coming to the second one, it states the beta anomer of D-glucose is less stable because of the equatorial position of C1 hydroxyl group. Again, if in case of beta anomer, we can already see in the last thing that the beta anomer was present as, yes, the beta anomer was present in 64%. If the beta anomer was less stable, then it would have not existed as in the beta form for 64%, because, one second, let me just say it again. So in case of glucose, we have this alpha form and is beta form. This beta form was 64% and alpha form was 36%. Now if beta form was unstable, it would have never existed. It is more percentage, it would have existed in less percentage. So obviously the second statement where it is stating that it is less stable because of the equatorial position of C1 hydroxyl group is a wrong statement. Because of its position as equatorial hydroxyl group, it should be more stable, because we all know placing the hydroxyl group at the equatorial position decreases the steric hindrance. So that will lead to more stability, not a distability in the structure. So the second statement is a false statement. The third statement says interconversions occurs due to the transient form of open chain form. In the last slide we showed that their interconversion occurs through this form, through this open chain form of D-glucose. So yes, the third statement is a true statement. And the fourth one says the relation between the two anomers are determined by their relative stability. Yes, absolutely. The relation between them is determined their stability. Because beta is more stable, it is remaining as 64% in the solution and the alpha is remaining as 36%. So fourth option is also a true statement. So we had to identify the false one. So only option 2 or the statements 2 is false. So the correct option for question number 19 should have been option B. Okay. Moving to the next one. Choose the correct denotation of the below aldohexoses and its epimers. So there are three structures A, B and C. You have to choose the correct options for them. So what is epimer we have discussed. So in case of epimers, we just change the stereochemistry of one carbon center. So that is why we call them epimers. So among these three structure, which one is very easy for all of you to identify? This one, this B1, because it is nothing but the structure of glucose. What is this? This is a D-glucose, YD. So this is your aldehyde group here, and the farthest, this is your first position aldehyde group. 2, 3, 4, 5, 6. So this is the farthest chiral carbon, and that has the hydroxyl group on the right hand side. So this is a D-glucose structure. You have to remember this structure. So we already have identified that B is our D-glucose. Now let us look at the other structure. So coming to the first one. So what is the difference between B and A? Where you can see the change? The only change between them is here at this position. This carbon center has been changed. See, in all the cases, let me mark here again. 1, 2, 3, 4, 5, 6. So 6 position is same, 5 position is also same, 4 is same, 3 is also same, but we can see a change in stereochemistry at the C2 position, right? So A should be a C2 epimer of D-glucose, B is already D-glucose we know, so A is a C2 epimer of D-glucose. So what is the C2 epimer of D-glucose? It is D-mannose. So this should have been option. This should have been A, D-mannose, C2 epimer of D-glucose. Now let us look at the structure for C. Again let us do the marking first. 1, 2, 3, 4, 5, 6. So one position is same, 2 is also same, 3 is same. We can see a change in this fourth carbon atom, a stereochemistry change between B and C in the fourth carbon atom. 5 and 6 are again same. So C should be a C4 epimer of B. So C4 epimer of D-glucose is D-galactose. So the correct option for question number 20 should have been option number A. A is D-mannose, which is a C2 epimer of D-glucose. B is D-glucose itself, and C is D-galactose, which is a C4 epimer of D-glucose. Hope this is clear. Let us move to the next one. So it is saying a structural representation of cellulose is shown below. Cellulose means it's the cell walls structural carbohydrate that is found in the cell walls of plants. So choose the correct answer. The bond that is joining the monosaccharide units is what kind of glycosidic bond? You have to understand. We can already see that it is a O glycosidic bond. But what kind of glycosidic bond it is? Please do write your answers in there because it is very important question. So these are the two units that are joined together. You have to identify the type of this glycosidic bond. So how do we name any kind of glycosidic bond? Yes, we have one answer. So first let us do the marking. So this one is our 6th, this is 5, 4, 3, I think I missed one. Yes, I have missed one. Sorry for that. 6, 5. Let me change the color. Then it will be easy for you to understand. In color this. Okay. So this was our 6th carbon atom. I missed this one. This is 5. This is 4. This is 3. This is 2. This is 1. Again, let us mark the second monosaccharide as well. This is 6. This is 5. This is 4. This is 3. This is 2. This is 1. So first of all, the linkage is between C1 and C4. So what kind of linkage it is? It is a 1,4 linkage. That is very obvious. And whether it is alpha or beta, how do we determine? So for determination of what? We do not require this second part at all. We just need to look at the first monosaccharide that we are dealing with. So this is our first monosaccharide, and in its case both the CH2OH group and this OH group are in the same direction. So CH2OH group is also in upward direction. So O, and this bond is also in, or your glycosidic bond is also in upward direction. You can see from here. This is upward direction. This is also in upward direction. So both are in same direction, that means it is cis, that means it is a beta bond. So it is a beta 1,4 glycosidic bond. So the correct option for this should have been option number D. Yes, absolutely correct. Correct answer. It is option number D. It is not alpha. It is, first do the markings, see which carbon atoms are linked to each other, then focus on the first monosaccharide, see whether it is alpha or beta, and name the glycosidic bond accordingly. Moving to next one. This one we all know, so we will not spend much time in it. It says, which structure among the following is the correct representation for D-glucopyranose in Haworth projection? So obviously we all know we have multiple times drawn the structure for glucose. So D-glucopyranose or D-glucose is actually the first one. The A1 is the correct structure for D-glucose. You have to remember it anyway, so I am not spending it much time. So, okay. This is the next question. It states, match the carbohydrates with their corresponding structural features. So we have three carbohydrates: mannose, fructose and galactose. We have to identify their structural features. So first of all, we all know what is mannose. Just 2 slides back we discussed this, that mannose, let me go back. Yes, just in this question we discussed that D-mannose was a C2 epimer of D-glucose and D-galactose was a C4 epimer of D-glucose. So let us go back to this question. So mannose is nothing but a C2 epimer of glucose and galactose is a C4 epimer of glucose. And what is fructose? At the beginning of this session we did this question. Fructose is a aldopentose. So we have our answers. So A goes with 3, B with 2 and C goes for 1. So the only option I think that matches is, yes, option number C. A is mannose, which is a C2 epimer of glucose. B is 2, that is fructose is the aldopentose, and C, that is galactose, is a C4 epimer of glucose. So the correct option for question number 23 should have been C. Okay. This is the next question. It states, match the monosaccharide column to the corresponding characteristic of or their usage column. So in the left hand side you have the monosaccharides and on the right hand side you either have their characteristic or their usage. You have to match them in pairs. Do that, take some time and select the correct option, then I will discuss it one by one. Try it by yourself. This is a very easy one, then I will do it one by one. Okay, I already have an answer. Yes, absolutely correct. So let us see. The first one is D-glyceraldehyde. Think some problem. No, it is fine. Okay. So the first monosaccharide that we are talking about is D-glyceraldehyde. So D-glyceraldehyde was the, or glycerol was the parent compound for carbohydrates, and from that we got our glyceraldehyde, which was the basic for the aldose sugars. So D-glyceraldehyde is one of the simplest sugars that is possible. So D-glyceraldehyde, let me mark here, 1 is matching with E. Next option is D-glucose. So D-glucose is something that is very, we all know that glucose is very important for our diet, because breakdown of glucose will give us the energy that we need for many of the bodily functions to occur. Next is D-fructose. Fructose, we all know it is C, the sweetest of all sugar, that is D-fructose. Next one is D-galactose. Galactose is something that is a part of a milk sugar. And at the last we have our D-ribose. Ribose sugar, obviously we know it is used in RNA. Then in the name itself it has ribonucleic acid, that means it has a ribose sugar. That means this monosaccharide is involved in case of RNA. So we have matched everyone, and the only option that matches with it is option number B. Yeah. The next question says, identify the conformations A and B of cyclohexane backbone state, which conformer is more stable and phi. So this is the four options that is shown here, A and B. Obviously you know the first one is A is a boat conformation and B is a chair conformation. From the structure the name is very clear. A1 is boat and the B1 is chair. So we can already discard option A and option D. Between B and C we know the correct option will happen. I can see one answer. Okay. And we know the chair conformation is much more stable than the boat conformation, because in case of boat conformation, whenever you put functional groups here in the axial position, there will be a steric repulsion between them. So in case of chair conformation, they are much more stable because this steric hindrance is much less in this chair form. So B form is much stable due to lesser steric crowding. So yes, absolutely the correct option should have been option B. A is boat, B is chair, and B is more stable due to lesser steric crowding. I think this is the last question. Okay. So it states, identify the product P obtained in the scheme shown. Name the reagent that is used to obtain P, which can also serve as a test for reducing sugars. So these are your options. You have your beta D glucose, and the open chain structure of beta D glucose is shown here as D glucose. Now from D glucose you have the formation of this compound called P. You have to identify the name of this compound P and the reagent that is being used here for this conversion. You all know that. Still, let me wait for some time for all of you to answer, then I will tell you. Which reagent is this and what is the name for this structure? Yes, we already have the answer, absolutely correct. So this structure is called D-gluconate. Why so? Because here you can see the CHO group or the aldehyde group has been converted to COO minus, which is known as a onate. So that means it is a glucose plus a COO minus, you are adding a onate. That means it is a D-gluconate, and the reagent that has been used here is nothing but Felling's reagent, because if you do remember the reaction, it was something like this. Is RCHO plus 2 copper 2 plus 5 OH minus in presence of alkaline medium. You have the oxidation of this aldehyde groups to RCOO minus, and you have Cu2O and water, and this Cu2O is responsible for giving a red coloration which confirms the existence of a reducing sugar or a aldose type of sugar, that can be glucose or any other aldose sugar, but the formation of this cuprous oxide confirms the presence of a aldose or a reducing sugar. So it is called as Felling's reagent, and ultimately the correct option should be P is T-gluconate and the reagent used is Felling's reagent. So yes, I think this is it. Okay, so we'll stop today's session here. Next day we'll do the week 11 lecture session. So thank you very much for joining.